Ancient DNA has revealed something deeply unsettling about our past. Not a new migration route, not a forgotten human species, but a moment when our own lineage came dangerously close to disappearing, so close that, for thousands of years, humanity was reduced to a population smaller than many modern villages. This was not a dramatic extinction event marked by ash layers or abandoned landscapes. There was no single catastrophe that ancient people could have pointed to and said, "This is the end." Life continued. Camps were rebuilt. Children were born. And yet, genetically, humanity was collapsing inward, shrinking to a narrow thread that almost snapped. For most of human history research, no one noticed. Archaeology could not see it. Fossils were too sparse, too scattered. Stone tools did not suddenly vanish. There was no obvious break in culture that screamed near extinction. To anyone looking only at the ground, humans appeared to endure as they always had. But DNA does not lie in the same way artifacts can mislead. When geneticists began comparing entire genomes, modern humans, alongside ancient remains, they found something impossible to ignore. Across all living humans today, genetic diversity is shockingly low, far lower than in many other species that survived the same climatic chaos, far lower than expected for a species that supposedly thrived for hundreds of thousands of years. At first, this seemed like a technical anomaly. Later, it became a pattern, and eventually, it became a timeline. The data pointed to a prolonged genetic bottleneck deep in the past, lasting not generations, but thousands of years. A period when Homo sapiens did not expand, did not diversify, and did not flourish. A period when survival itself became the only story. What makes this revelation so striking is not just how close we came to extinction, but how quietly it happened. No legends remember it. No myths preserve it. Even our bones do not clearly record it. Only DNA, passed down cell by cell, still carries the signal. This story is not about drama or mystery. It is about limits, biological, environmental, and demographic. And it begins with what we thought we knew, before the genetic evidence forced us to look again. Before ancient DNA entered the picture, the story of early Homo sapiens was built almost entirely from what could be seen and touched. Fossils, stone tools, cut marks on bones, and the slow accumulation of sites across Africa and Eurasia formed the foundation of human prehistory. From this evidence, researchers reconstructed a narrative that appeared coherent and reassuring. According to this traditional view, Homo sapiens emerged in Africa roughly 300,000 years ago. Early populations were small, but stable. They occupied diverse environments, from open savannas to wooded regions and coastal zones. Over time, they developed increasingly flexible behaviors, new tool types, broader diets, and eventually symbolic expression. These traits were interpreted as signs of long-term demographic health. When climate shifted, humans adapted. When environments dried or cooled, populations moved. Archaeological layers showed continuity rather than collapse. Stone tool traditions changed gradually, not abruptly. Campsites reappeared again and again in the same regions, suggesting persistence rather than abandonment. To archaeologists, this pattern implied survival through resilience. Even the harshest periods of the Pleistocene ice ages were framed this way. Humans were thought to contract into refuges during colder, drier phases, then expand again when conditions improved. These contractions were assumed to be temporary and limited. Population sizes may have dipped, but extinction-level threats were considered unlikely. Comparisons with other human species reinforced this confidence. Neanderthals and other archaic humans disappeared, but Homo sapiens endured. The assumption was that our species possessed cognitive or social advantages that buffered it against extreme risk. Survival seemed built into the system. Importantly, there was no obvious archaeological signal pointing to a near extinction, no long, continent-wide gaps in occupation, no sudden loss of technology across all regions, no clear moment where humans appeared to vanish and later reappear. The material record suggested continuity, even if unevenly distributed. This shaped how researchers thought about population size. Numbers were estimated indirectly, based on site density and geographic spread. If tools were found across wide areas, populations were assumed to be large enough to sustain them. Sparse evidence was often explained by preservation bias rather than demographic collapse. In this framework, genetic diversity was rarely part of the discussion. DNA from ancient remains was either unavailable or too degraded to analyze. Modern human genetic variation was known to be relatively low compared to some species, but this was often attributed to recent expansions out of Africa or to cultural factors, not deep time crises. The idea that Homo sapiens might have been reduced to only a few thousand individuals for an extended period simply did not fit the visible evidence. It conflicted with the apparent geographic spread of early humans. It conflicted with the continuity of stone tool traditions, and it conflicted with the assumption that a species capable of such wide dispersal must have been demographically secure. As a result, models of human evolution emphasized growth, expansion, and innovation. Bottlenecks were acknowledged, but treated as short-lived and localized. The deeper past was imagined as a slow build-up toward success, not a prolonged struggle to avoid disappearance. What was missing from this picture was a direct measure of population size and connectivity over time. Archaeology could show where people had been, but not how many there were, or how isolated they might have become. Fossils could reveal anatomy, but not genetic variation across entire populations. This gap left room for assumptions to harden into consensus. Humans survived, therefore humans must always have been numerous enough to survive. The absence of evidence for collapse was taken as evidence of absence. That confidence would begin to erode only when researchers learned to read a different archive of the past, one not shaped by erosion, chance discovery, or incomplete excavation, but carried silently within the cells of every living human today. The shift began quietly, in laboratories rather than excavation sites. For decades, DNA older than a few thousand years was assumed to be unreadable. Heat, moisture, and time break genetic material into fragments so small they resemble dust. Early attempts at recovery were plagued by contamination and false signals. Many researchers believed that the deep human past would remain genetically inaccessible. That assumption changed in the late 20th and early 21st centuries as methods improved. Clean room laboratories reduced contamination. New chemical techniques allowed scientists to extract and amplify tiny pieces of degraded DNA. High-throughput sequencing made it possible to read millions of fragments at once and reassemble them statistically, rather than relying on intact strands. The first successes came from relatively recent remains in cold environments. Bones preserved in permafrost or caves yielded DNA tens of thousands of years old. From these samples, researchers reconstructed partial genomes of extinct human groups. What began as proof of concept soon became a powerful tool. But the most transformative insight did not come from any single fossil. It came from comparing genomes across time. When ancient genomes were aligned with those of living humans, patterns emerged that could not be explained by archaeology alone. Sections of DNA showed unexpected uniformity. Across populations separated by continents, genetic variation appeared compressed, as if much of humanity shared a recent and very small ancestral pool. To test this, geneticists turned to population modeling. By measuring how often genetic mutations occur and how they spread through populations, it is possible to estimate effective population size over time. This does not count individuals directly. Instead, it reflects how many people were contributing genetically to future generations. When these models were applied to whole genome data from diverse modern populations, the results converged on a striking conclusion. Somewhere deep in the Middle Pleistocene, long before the major migrations out of Africa, effective population size dropped dramatically. Not briefly, not locally, but globally, and for an extended period. The signal suggested a bottleneck lasting tens of thousands of years, beginning roughly 900,000 years ago and extending for a vast stretch of time. During this interval, the effective breeding population of ancestral humans may have fallen to only a few thousand individuals. This was not inferred from a single method. Different analytical approaches, using independent datasets, pointed in the same direction. Coalescent models, which trace genetic lineages backward in time, showed long periods where lineages failed to branch. Mutation patterns clustered tightly. Genetic diversity failed to recover for an unusually long duration. Crucially, this bottleneck did not align neatly with previously emphasized events. It did not correspond to a known mass extinction horizon in the fossil record. It did not coincide with a sudden technological collapse. Instead, it appeared to overlap with a period of profound climatic instability, when glacial cycles intensified and environmental conditions fluctuated rapidly. Ancient DNA also helped clarify something archaeology could not easily resolve: connectivity. Even if humans occupied wide geographic areas, they may have been isolated into small, fragmented groups with little gene flow between them. From a genetic perspective, isolation can be just as dangerous as low numbers. Additional support came from comparisons with other species. Many animals that survived the same climatic periods retained higher genetic diversity than modern humans. This suggested that whatever affected early Homo lineages was unusually severe or prolonged. As more ancient genomes were sequenced, including those from archaic humans, the picture sharpened. The bottleneck appeared to predate the divergence of several later human groups. In other words, it affected a common ancestral population long before the emergence of Neanderthals, Denisovans, or fully modern Homo sapiens. This reframed the role of ancient DNA entirely. Instead of simply identifying who interbred with whom, genetics began to reveal when almost no one was left. The implications were profound. A species thought to be steadily expanding was shown to have endured a demographic collapse so deep that it permanently shaped our genetic landscape. The narrow range of variation seen in humans today was not just the result of recent migrations. It was the echo of survival at the edge of extinction. And yet, despite the severity of this bottleneck, the archaeological record remained largely continuous. Tools persisted, sites were reused, cultural traditions endured. This disconnect forced researchers to confront a difficult truth. Material continuity does not guarantee demographic security. Ancient DNA had exposed a hidden chapter of human history, one that could not be seen in stone or bone alone. a chapter defined not by innovation or expansion, but by endurance under extreme constraint. What the genetic data ultimately revealed was not just a decline in numbers, but a fundamental reshaping of humanity's future. And that reshaping would only become clear once researchers examined exactly what survived and what was lost during that long, quiet crisis. What emerged from the genetic evidence was not a single dramatic event, but a long, grinding constriction of humanity's biological future. The bottleneck revealed by ancient DNA was severe not only because of how small the population became, but because of how long it remained that way. For tens of thousands of years, ancestral human populations existed in a state of extreme demographic vulnerability. In genetic terms, this meant that many lineages simply disappeared. When populations shrink to very low numbers, chance begins to dominate inheritance. Entire branches of genetic variation can vanish if the individuals carrying them fail to reproduce. Over time, this produces a population that appears outwardly stable, but internally stripped of diversity. The DNA shows exactly this pattern. Large portions of genetic variation that would be expected in a long-lived, widespread species are missing. Instead, modern humans share a surprisingly narrow set of ancestral lineages. This is not the signature of gradual refinement. It is the signature of survival through elimination. One of the most striking findings is how this bottleneck reshaped later human evolution. Because the ancestral population was so small, any traits carried by those few survivors became disproportionately important. Genetic quirks, vulnerabilities, and strengths alike were amplified simply because there were no alternatives left. This helps explain why humans today are genetically more similar to one another than many other mammals. Two people from opposite sides of the planet are, at the DNA level, remarkably alike. The bottleneck compressed our species into a tight genetic corridor, and everything that came afterward passed through that narrow opening. The timing of this collapse also challenged long-standing assumptions. It occurred far earlier than the emergence of Homo sapiens as a distinct species. This means the crisis was not faced by modern humans in the cultural sense, but by ancestral populations from which several human lineages would later diverge. In effect, the bottleneck acted as a filter. Only a small subset of the broader human family survived it. Afterward, those survivors split into separate evolutionary paths, eventually giving rise to groups like Neanderthals, Denisovans, and later Homo sapiens. But all of them inherited the same reduced genetic foundation. This reframes extinction in a crucial way. Neanderthals and Denisovans did not disappear because they were uniquely fragile. They emerged from an already constrained lineage. Their later extinctions occurred against a backdrop of limited genetic resilience inherited from that earlier crisis. Ancient DNA also revealed that recovery from the bottleneck was slow. Population size did not rebound quickly once conditions improved. Genetic diversity accumulated at a cautious pace, suggesting that human groups remained small, scattered, and intermittently connected long after the worst period had passed. This prolonged fragility increases the likelihood that environmental stressors played a central role. During this time, Earth experienced intensified glacial cycles, shifts in rainfall patterns, and changes in vegetation that would have disrupted food sources across Africa and Eurasia. Even without a single catastrophic event, sustained instability could keep populations trapped at low numbers. Importantly, the genetic data does not point to a complete collapse everywhere at once. Instead, it suggests fragmentation. Small groups survived in pockets, isolated from one another for long periods. From an archaeological perspective, this can still look like continuity. From a genetic perspective, it is a near extinction stretched across millennia. Another critical insight concerns interbreeding. Later, genetic exchanges between human groups, once seen as rare or exceptional, take on new meaning in light of the bottleneck. When diversity is low, interbreeding becomes a mechanism of survival, restoring some variation that had been lost. This may help explain why gene flow between human populations persisted whenever contact was possible. The DNA also clarifies why certain traits appear suddenly widespread in later humans. These were not necessarily rapid innovations. Many may have existed earlier, but only became universal after the bottleneck removed competing variations. What looks like sudden emergence can, in reality, be the result of survival through constraint. Perhaps the most sobering implication is how invisible this crisis was without genetic evidence. A species can hover on the edge of extinction without leaving obvious scars in tools or habitats. Cultural persistence can mask biological peril. Humans continue to make tools, occupy landscapes, and pass knowledge across generations, even as their genetic future narrowed dramatically. In this sense, ancient DNA did more than revise population estimates. It revealed a fundamental mismatch between appearance and reality. Humanity looked stable while becoming dangerously small. The archaeological record told a story of endurance, while the genome told a story of survival against overwhelming odds. This discovery forces a reconsideration of what it means to persist. Survival is not always expansion. Sometimes it is simply not disappearing. And in that long interval, revealed by DNA, that was all humanity managed to do. The consequences of this hidden bottleneck reach far beyond population numbers. They reshape how we understand what it means to be human, not in abstract terms, but in biological reality. Every living person today carries the imprint of that ancient narrowing. Our shared genetic similarities are not just a reflection of recent history, but the outcome of a much older survival constraint. This helps explain why humanity, despite its global presence, is genetically young. The differences between populations are shallow, layered on top of a deep uniformity. Skin color, facial features, and other visible traits vary. But beneath them lies a remarkably consistent genetic framework. Ancient DNA shows that this consistency is not accidental. It is inherited from a time when variation itself was a liability that could not be sustained. The bottleneck also reframes how we think about adaptability. Humans are often described as uniquely flexible, capable of thriving in almost any environment. The genetic evidence suggests a more nuanced picture. Adaptability did not prevent near extinction. It emerged after it. Many of the traits associated with later human success may have been shaped, refined, or amplified precisely because only certain combinations survived that long period of scarcity. This perspective brings humility to the story of human evolution. Survival was not guaranteed. Intelligence alone was not a safeguard. Sociality, technology, and learning mattered, but they operated within severe biological limits. The lineage that endured did so not because it was destined to succeed, but because it managed to persist when success was not an option. Ancient DNA also alters how we view later expansions. When humans eventually spread widely across continents, they did so from a constrained genetic base. Each migration carried only a fraction of an already reduced diversity. This means that the story of human dispersal is also a story of repeated narrowing, layered upon an older, deeper bottleneck. This has practical implications for how scientists interpret disease susceptibility, genetic disorders, and immune responses in modern populations. Some vulnerabilities may trace back not to recent history, but to the limited genetic toolkit that survived the ancient collapse. The past is not just informative, it is present. At the same time, the bottleneck underscores a powerful shared inheritance. Every modern human, regardless of ancestry, descends from the same small group of survivors. There is no population on Earth untouched by this history. Differences that appear large on the surface are minor when set against the profound genetic commonality revealed by ancient DNA. This does not erase cultural or historical distinctions, but it grounds them in a biological context of shared origin. Humanity is not a collection of separate lineages that later converged. It is a single lineage that almost failed, then diversified cautiously from what remained. Importantly, this understanding remains firmly rooted in evidence. Ancient DNA does not tell a story of triumph or destiny. It documents survival under constraint, followed by gradual recovery. It shows how chance, environment, and inheritance intersected in ways that no individual or group could control. By revealing this hidden chapter, genetics adds depth to the human story without embellishment. It replaces assumptions of steady progress with a more fragile, but more accurate, account. Humanity endured not because extinction was impossible, but because, against considerable odds, it did not happen. For most of our history, the limits of human survival were invisible. Stone tools endured. Camps were rebuilt. Landscapes were reoccupied. From the outside, humanity appeared continuous and secure. What ancient DNA revealed is that this appearance was misleading. Deep in the past, long before written history or recognizable cultures, our lineage passed through a period of extreme constraint. Population size narrowed. Genetic variation collapsed. For thousands of years, humanity existed in a fragile state, sustained not by abundance or expansion, but by persistence alone. This was not a moment of progress. It was a moment of survival. Ancient DNA matters because it captures what archaeology cannot easily see. It records absence as clearly as presence. It preserves the loss of lineages, the silence of genetic branches that ended without descendants. Through this record, we can now recognize that the modern human genome is not simply the product of recent migrations or cultural success, but the outcome of an ancient demographic crisis that shaped everything that followed. This knowledge does not diminish the human story. It grounds it. It shows that resilience is not the same as invulnerability, and that continuity can coexist with extreme risk. The narrow genetic foundation shared by all people today is not evidence of destiny, but of endurance under conditions that could easily have led to extinction. Ancient DNA continues to refine this picture as new methods and discoveries emerge. With each improvement, the past becomes less abstract and more precise, not louder or more dramatic, but clearer. Humanity's survival was not guaranteed, and it left a measurable imprint that still defines us. The story revealed by ancient DNA is quiet, but it is fundamental. It reminds us that the human past is not only written in tools and bones, but in the shared genetic legacy carried by every living person.